In the quiet hours, when the world slows and the rhythms of life settle into a gentle hum, there is another rhythm at work, one far too small to see, yet endlessly intricate. It is the rhythm of chemistry inside living things, a ceaseless dance of molecules, of atoms joining and parting, of delicate exchanges that have been unfolding for billions of years. This is the world of biochemistry, the study of life at the molecular level. It is a science that listens closely to the whispers of the smallest components of existence, finding meaning in the way they meet, interact, and transform. Every living cell, no matter how tiny or complex, is a miniature chemical world. Within its boundaries, thousands of reactions take place in perfect coordination. Sugars are broken down for energy. Proteins are folded with care. Signals pass softly from one molecule to the next, like messages passed in a great silent hall. And all of it, every intricate movement, follows the laws of chemistry. Biochemistry is a bridge between biology, the study of life, and chemistry, the study of matter. It is where the two meet and speak the same language. Here, life is not seen as a mystery to be solved suddenly, but as a long, unfolding pattern where each answer leads softly to the next. This pattern connects all living things, from the green leaf drawing in sunlight, to the fish gliding through a stream, to the quiet breath rising and falling in your chest. Though the details differ, the molecules are the same. The energy flows in the same gentle currents. The chemistry is universal. Even the most ordinary moments, the slow beat of your heart, the way your muscles move when you turn in bed, the simple act of breathing in and out, are guided by a hidden network of biochemical reactions. In this way, biochemistry is both invisible and everywhere. It can feel remarkable to think that you are, in a sense, a collection of carefully arranged molecules. Yet that arrangement is not random. It is purposeful, precise, and sustained by a constant exchange of matter and energy. This is the quiet truth of living things. They are not static objects, but flowing systems, always in motion at the molecular level. Inside you, a single heartbeat is accompanied by countless chemical changes. Molecules of glucose are broken apart to release energy, a process that has been occurring since the earliest life forms first emerged on Earth. The very same oxygen you breathe in travels through the bloodstream and slips into your cells, where it helps to complete this ancient process. The food you eat, whether fruit, grain, or protein, is not simply absorbed whole. Instead, it is taken apart in slow, careful steps. Large molecules are broken into smaller ones, then reassembled into forms your body can use. Each transformation is guided by enzymes, the gentle catalysts of biochemistry, who nudge reactions forward without forcing them. Water, too, is everywhere in this microscopic world. It is the silent stage on which all biochemical reactions occur. Every cell is a tiny chamber of water, filled with dissolved salts, sugars, and proteins. Without water's remarkable properties, its ability to dissolve so many substances, its capacity to absorb heat without changing temperature quickly, the delicate balance of life's chemistry would collapse. Water molecules are constantly in motion, flowing into and out of cells, shaping their volume, carrying nutrients to where they are needed, and removing waste when it is no longer useful. This movement is so ordinary, so constant, that it is easy to forget it is happening at all. But without it, life could not persist for even a moment. If you were able to shrink down to the scale of a single molecule, the world would appear very different. You would see a crowded, dynamic space. Proteins would fold into complex shapes beside you. Lipid molecules would drift together, forming the flexible membranes that enclose each cell. Tiny strands of nucleic acids would curl and twist, carrying coded instructions written in a molecular language older than any human tongue. There would be no empty silence here, not because it is noisy, but because it is busy. Reactions would be taking place in every direction, yet not chaotically. Each has its place, its timing, and its purpose. The cell is like a city, with no wasted movement. Food arrives just where it is needed. Messages are sent without delay. Waste is carried away efficiently. And while the city of the cell is active, it is also harmonious. One process flows into the next. The breakdown of one molecule provides the starting material for another. Energy produced in one reaction is stored, then drawn upon by another, keeping the cycle unbroken. Biochemistry is the study of this harmony. It asks, what molecules are present in living things? How do they interact? What patterns do they follow? And, perhaps most beautifully, how do such patterns give rise to life itself? This is not the chemistry of explosions or sudden change. It is the chemistry of steady, ongoing transformation, a chemistry that unfolds in whispers, shaping everything we know as life. It is comforting to think that these same molecular processes are not unique to us. The chemical pathways in a tree leaf, in a hummingbird's wing, in the roots of grass, they are variations on the same themes that flow through our own cells. The glucose molecule that fuels a cell in your body is built in much the same way as the glucose molecule made in a green leaf by sunlight. The ATP molecule that carries energy in a bird's muscle is identical to the one in your own. This shared chemistry is a reminder that all living things, no matter how different they seem, are bound together by the same deep currents. As we begin this quiet exploration of biochemistry, we will see that these currents follow certain rules. They are shaped by the properties of atoms, by the bonds they form, and by the environments in which they move. We will see how small molecules join to create larger ones, and how these larger molecules fold, twist, and interact to form the machinery of life. We will learn how energy moves within a cell, how it is stored, and how it is used. We will look at the delicate balance between building up and breaking down the endless cycle of creation and transformation that sustains us. We will see how cells send messages, how they sense their surroundings, and how they respond to subtle changes in their internal chemistry. And we will reflect on how all of this, from the simplest chemical reaction to the most complex genetic process, is part of one vast and continuous pattern. So as we set out, it may be helpful to imagine biochemistry not as a subject to be hurried through, but as a landscape to move through slowly. A landscape where every molecule has a story, and every reaction is part of a greater whole. In this landscape, the pace is never frantic. Even the fastest biochemical reactions occur in the context of a life that is measured not in seconds alone, but in seasons, generations, and eons. These processes have been in motion since long before humans appeared, and they will continue long after. So breathe slowly. Let your mind rest in the thought that, even now, while you are still, your cells are quietly alive with activity. Energy is flowing, molecules are moving, and the chemistry of life carries on always, and without pause. This is where our journey begins, in the smallest spaces, with the quietest motions, in the delicate, constant chemistry of life. 2. Defining biochemistry. Biochemistry, at its heart, is the study of life's chemistry. It is the gentle meeting point where the principles of chemistry, the science of matter, join with the principles of biology, the science of living things. In this quiet space between the two disciplines, we begin to understand how the smallest particles of matter can come together to create something as vast, as complex, and as enduring as life. At first glance, chemistry and biology might seem like separate worlds. Chemistry deals with atoms, molecules, and the reactions between them. Biology speaks of cells, tissues, and entire organisms. But if we were to peel back the layers of a living system, molecule by molecule, the border between the two disappears. Life, after all, is chemistry in motion. When scientists speak of biochemistry, they speak of the chemical processes that make life possible. These processes are not confined to rare or unusual moments, they are happening constantly in every living being. The food you eat being broken down into molecules small enough to pass into your blood, the movement of oxygen from your lungs into your cells, the folding of a new protein in a cell's interior, the release of a hormone from a gland, carrying a signal to another part of the body. Each of these is a biochemical event, a transformation of matter and energy taking place within the setting of life, and each one follows the same physical and chemical laws that govern the non-living world. Biochemistry does not replace chemistry. It refines it. It asks, how do chemical principles operate in the delicate, crowded environment of a living cell? In the stillness of the laboratory, a chemical reaction might take place in a beaker of pure liquid. In a living organism, that same type of reaction unfolds in a space packed with thousands of other molecules, each one jostling and reacting, all within a structure smaller than the head of a pin. This is part of the beauty of biochemistry. The same rules apply, but life has found ways to use them in the most intricate and coordinated fashion. Biochemistry also does not replace biology. It deepens it. If biology tells us what living things do, biochemistry tells us how they do it at the molecular level. Biology might say, a muscle contracts. Biochemistry explains, molecules of ATP, the cell's energy currency, are broken apart, releasing energy. That energy is transferred to protein filaments in muscle fibers, causing them to slide past one another and shorten the cell. Biology might say, a plant grows toward the light. Biochemistry explains, light-sensitive proteins detect the direction of sunlight, triggering the release of specific hormones. These hormones stimulate cell expansion on one side of the stem, gently turning the plant toward the source of light. In this way, biochemistry reveals that even the simplest biological action is built from countless, quiet, molecular events. It is important to understand that biochemistry is not concerned with life in the abstract. It is concerned with life as it is, in the form of molecules and reactions. This means that biochemistry touches every level of living systems, from the smallest building blocks of life to the grand patterns of metabolism that sustain an entire organism. We often speak of four major categories of molecules in biochemistry: carbohydrates, which store and provide energy and sometimes serve as structural elements; lipids, which form cell membranes and store energy in dense, compact forms; proteins, the versatile molecules that catalyze reactions, carry signals, and form the structural framework of cells; and nucleic acids, which store genetic information and direct the synthesis of proteins. Each of these categories has its own story, a story of how its molecules are built, how they interact, and how they take part in the cycles of life. In time, we will visit each of them in turn. Biochemistry is also a science of patterns and pathways. In a living cell, no chemical reaction stands alone. The breakdown of a sugar molecule feeds into the production of ATP. The breakdown of ATP powers the work of proteins. Proteins in turn make possible the transport of sugars into the cell, starting the process over again. This interconnectedness means that biochemistry is not just a study of things, but of flows. It is the study of how matter and energy move through the molecular machinery of life, how they are transformed and conserved, and how they keep the organism balanced in the face of constant change. One of the most important ideas in biochemistry is the concept of homeostasis, the tendency of living things to maintain stable internal conditions. This stability is not rigid, it is dynamic. A living system must adjust constantly to changes in its surroundings, whether those changes are sudden, like a drop in temperature, or gradual, like the slow decline in available nutrients. Biochemistry helps us understand the molecular tools life uses to make these adjustments: enzymes that speed or slow reactions, hormones that carry signals across the body, membrane channels that open or close to let substances pass. The goal is always the same: to keep the cell, and therefore the organism, in a state where life can continue. We might also think of biochemistry as a kind of translation. It translates the language of molecules into the language of life. It shows us how the arrangement of atoms in a small molecule can determine the behavior of an entire organism. A subtle change in the shape of a protein can mean the difference between a healthy cell and one that cannot function. A single mutation in DNA can alter the structure of a vital enzyme, affecting the flow of an entire metabolic pathway. In this sense, biochemistry helps bridge the vast distance between the microscopic and the macroscopic, between the realm of molecules and the world we experience with our senses. It is also a science with practical importance. Every field of medicine rests, in part, on biochemical knowledge. Understanding how enzymes work helps scientists design drugs that can block or enhance specific reactions. Knowing how cells produce energy guides the treatment of metabolic disorders. Recognizing the chemical signals that pass between cells opens doors to therapies for diseases of the nervous and endocrine systems. Agriculture, nutrition, environmental science, all are shaped by biochemistry. Even our understanding of life beyond Earth would begin with questions of biochemistry. Could molecules like those on Earth exist elsewhere? Could they assemble into something that breathes, grows, and changes? And yet, for all its breadth and depth, biochemistry is not an overwhelming subject when approached gently. Its concepts build naturally on one another. The shapes of molecules lead to the functions they perform. The functions of molecules lead to the behaviors of cells. The behaviors of cells lead to the life of the organism. It is a chain, and each link is made of the same material, the quiet chemistry of life. So, when we define biochemistry, we are defining more than a branch of science. We are naming the study of how life works at its smallest scale, how atoms come together to form molecules, how molecules interact to build the architecture of cells, how cells coordinate their chemistry to sustain the ongoing miracle we call living. It is the study of the constant conversation between matter and life, a conversation that has been speaking in the same elemental language for billions of years. In the next part of our journey, we will turn to the simplest question of all: what is life made of? We will explore the chemical elements that form the foundation of every living thing, from the smallest bacterium to the largest tree, and of course, to us. 3. Historical roots of biochemistry. The story of biochemistry is older than the name itself. Long before the word existed, people were unknowingly observing and using biochemical processes in daily life. They brewed beer from grains, baked bread with yeast, and fermented fruit into wine. They preserved food with salt, extracted dyes from plants, and tanned hides for clothing. All of these were the results of chemical transformations within living or once living materials, though no one yet knew the molecular details. It would be centuries before anyone began to understand that these changes were not the work of chance or magic, but the result of consistent, repeatable processes. Slowly, human curiosity moved from simply using these processes to asking why they worked at all. In ancient Greece, philosophers like Aristotle and Hippocrates speculated on the nature of life and health. They spoke in terms of balance, the balance of the four elements, or the four bodily humors, without the benefit of modern chemistry. These early ideas were more poetic than precise, yet they shared a key insight. Living things depend on an internal harmony to survive. In a way, they were sensing the earliest outline of what we now call homeostasis, though the underlying molecular details were far beyond their reach. The turning point began with the rise of modern chemistry in the 17th and 18th centuries. Scientists learned to measure, to weigh, and to test the substances they worked with. They discovered that matter was made of elements, that reactions could be explained in terms of rearranged particles rather than mystical forces. This was the era when chemistry began to take its modern shape, and it opened the door to looking at life through a chemical lens. By the late 18th century, a few scientists began to suspect that living processes could be understood in terms of chemistry. One important figure was Antoine Lavoisier, often called the father of modern chemistry. He studied respiration, the act of breathing, and discovered that it was, in essence, a form of slow combustion. Oxygen from the air combined with carbon in the body to release energy and produce carbon dioxide. It was one of the first clear demonstrations that a process inside a living body could be explained by the same chemical principles that governed a flame. Through the 19th century, biochemistry began to emerge as its own discipline. The study of fermentation became a central puzzle. Brewers had long known that yeast could turn sugar into alcohol, but no one understood how. Some scientists believed fermentation required a vital force found only in living organisms. Others began to suspect that the process might be driven by specific substances within the yeast itself. The answer came gradually. In 1833, two French chemists, Anselme Payen and Jean-François Perceau, discovered a substance from plants that could break down starch into sugar. They called it diastase, the first enzyme ever identified. Enzymes would later become one of the central topics of biochemistry, but at the time, this was only the beginning. Later in the century, Louis Pasteur demonstrated that fermentation was carried out by living microorganisms. He famously declared that fermentation is life without air. This supported the idea that chemical transformations in food and drink were driven by the activities of living cells. Pasteur believed enzymes could not work outside of living cells, but even he did not yet see the full picture. That picture became clearer at the turn of the 20th century, when Eduard Buchner, a German chemist, made a groundbreaking discovery. He showed that yeast extracts, without any living yeast cells present, could still carry out fermentation. This meant that the chemical machinery of life could be separated from the cells themselves and studied in isolation. It was a revelation. It marked the moment when biochemistry could truly begin to study life's chemistry directly, without the barrier of the whole living organism. With this discovery, biochemistry moved rapidly forward. Scientists began isolating enzymes, identifying vitamins, and studying how the body uses carbohydrates, proteins, and fats. They mapped the pathways by which food becomes energy, and energy is transformed into the building blocks of life. In the mid-20th century came perhaps the most famous moment in modern biochemistry, the discovery of the structure of DNA in 1953 by James Watson and Francis Crick, building on crucial X-ray data from Rosalind Franklin and Maurice Wilkins. DNA's double helix form revealed the elegant way genetic information is stored and copied, a molecular script passed from one generation to the next. It was a bridge between chemistry and biology so complete that it forever changed both fields. From there, biochemistry expanded into molecular biology, genetics, and biotechnology. The tools of the field became more refined. Ultracentrifuges to separate molecules by weight, spectrophotometers to measure their light absorption, electron microscopes to see their shapes. Researchers could now observe, in increasing detail, the precise dance of molecules that makes life possible. Today, biochemistry touches nearly every area of science and medicine. It guides the design of new medicines, informs nutrition research, helps us understand diseases at the molecular level, and even shapes our search for life beyond Earth. Yet, at its core, the essence of biochemistry remains unchanged from its earliest days: to understand the chemistry that underlies living things. Knowing its history is more than an exercise in curiosity. It reminds us that our current understanding is the result of countless careful observations, gradual improvements in tools and methods, and the work of many minds across centuries. The science did not arrive fully formed. It grew slowly, one question at a time, each answer leading to the next. And so, as we look back, we can see the quiet arc of discovery, from ancient ideas about life's hidden forces to the recognition that living processes are, at their heart, chemical in nature, to the detailed understanding of molecules and pathways we have today. In our next step, we will begin to explore the most basic chemical ingredients of life. These are the elements that make up every living cell, the raw materials from which all of life's molecules are built. Just as a few letters can form countless words, a small set of elements can be arranged into the vast diversity of life. 4. The chemical elements of life. When we think of life, we might picture the great variety of living forms: towering trees, darting fish, birds in flight, or ourselves in quiet reflection. Yet, beneath this endless variety, life is built from a surprisingly small set of basic materials. All living things, from the smallest bacterium to the largest whale, are made primarily from a handful of chemical elements. These elements are the true alphabet of life's chemistry. Of the many elements that exist in the universe, only a select few are found in large amounts in living systems. The 6 most important are carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur. Together, they make up the vast majority of the mass of living organisms. Though simple on their own, these elements can be arranged into countless combinations, forming the molecules that carry out every process of life. Carbon is at the heart of this system. It is the central letter in life's chemical alphabet, the foundation on which so many molecules are built. The reason is simple but profound. Carbon atoms can form 4 strong covalent bonds with other atoms, allowing them to link into chains, rings, and complex branching structures. No other element matches this versatility. Through these bonds, carbon creates the backbone for sugars, lipids, proteins, and nucleic acids, the 4 great families of biological molecules. It is carbon's ability to connect in so many ways that gives life its molecular richness. Hydrogen, the simplest of all elements, is everywhere in living things. Each hydrogen atom is a single proton, orbited by a single electron, yet it is essential to the structure of water, to the building of organic molecules, and to the transfer of energy within cells. It often forms bonds with carbon and oxygen, helping shape the structure of biomolecules. In the water molecule, hydrogen's partial positive charge allows it to form hydrogen bonds, the delicate attractions that hold water molecules together, giving water its unique life-sustaining properties. Oxygen is equally vital. In water, oxygen carries a partial negative charge, drawing hydrogen atoms toward it and creating the polarity that makes water such a powerful solvent. In respiration, oxygen is the final acceptor of electrons in the chain of reactions that extract energy from food. Without oxygen, most forms of complex life could not harvest enough energy to survive. Oxygen atoms also appear in carbohydrates, lipids, and many proteins, adding reactive sites that enable chemical change. Nitrogen is a cornerstone of proteins and nucleic acids. It is found in the amino groups that give amino acids their name, and in the nitrogenous bases that form the rungs of the DNA ladder. While nitrogen gas is abundant in Earth's atmosphere, its bonds are so strong that most organisms cannot use it directly. Instead, it must be fixed into reactive forms ammonia, nitrates, or nitrites by certain bacteria and archaea. Once fixed, nitrogen moves through the biosphere, feeding into the growth of plants, animals, and microbes alike. Phosphorus, though less abundant than the others, is essential for energy transfer and storage. It is part of ATP adenosine triphosphate, the molecule that carries chemical energy within cells. The breaking of phosphate bonds in ATP releases energy for cellular work. Phosphorus also forms the backbone of DNA and RNA, linking nucleotides into long, information-rich strands. In bones and teeth, phosphorus joins with calcium to create strong mineral structures. Sulfur is found in certain amino acids, notably cysteine and methionine, and in some vitamins. Its ability to form disulfide bonds helps stabilize the shapes of proteins, ensuring they hold the correct form to function. Though required only in small amounts, sulfur's role is critical to the stability of many molecules. Beyond these 6 core elements, other elements play supporting roles. Sodium, potassium, calcium, magnesium, chlorine, and iron are among the most important. They may be present in smaller amounts, but they are essential nonetheless. Sodium and potassium ions help create electrical signals in nerve and muscle cells. Calcium acts as both a structural component in bones and a messenger in signaling pathways. Magnesium often sits at the center of chlorophyll, the green pigment that captures light energy in plants. Iron, bound in the hemoglobin of red blood cells, carries oxygen from the lungs to the tissues. Even trace elements such as zinc, copper, manganese, iodine, and selenium are required for life. Their amounts are tiny, but without them, key enzymes could not function, hormones could not be made, and antioxidant systems could not protect cells from damage. In the grand view, all of life's molecules are constructed from this relatively short list of building blocks. What gives life its vast diversity is not the number of elements, but the infinite ways they can be joined. Just as all the words in a language can be formed from a limited alphabet, all the molecules in living systems are built from the same elemental set. The difference comes from arrangement, from sequence, and from the shape that the arrangement creates. These elements do not simply sit side by side. They interact. They share electrons to form covalent bonds, or attract each other through weaker hydrogen bonds and ionic bonds. They form the chains of carbohydrates, the rings of nucleotides, the folded shapes of proteins. They assemble into membranes that enclose cells, into fibers that give tissues strength, into molecular machines that carry out the work of life. It is humbling to realize that the same carbon in the cells of a leaf might, after the leaf decays, become part of the shell of a mollusk or the muscle of an animal. The same oxygen atom you breathe in today may one day be part of a sugar molecule in a tree. The elements are not fixed to one purpose. They move continually through the great cycles of Earth, the carbon cycle, the nitrogen cycle, the water cycle, taking on new roles in each form they inhabit. In this way, the chemistry of life is also the chemistry of the planet. We are not separate from the material of the Earth. We are part of it, shaped by the same elements, sustained by their continual movement and transformation. As we continue our journey, we will see how these elements combine into the 4 great families of biological molecules. We will see how structure and function are woven together, how the properties of atoms ripple outward to shape the behavior of cells, and how this simple elemental alphabet tells the entire story of life. 5. The unique role of water. If we could shrink ourselves down and look closely at the inner world of a living cell, we would find that it is not a dry, static place. It is filled with a clear, shimmering fluid, a moving, dynamic environment in which all the molecules of life drift, meet, and react. That fluid is water, and it is far more than just a background or a container. It is the medium of life, the stage on which every biochemical reaction unfolds. Water is so common that it is easy to overlook its importance. Yet in every living thing, from a simple bacterium to the human body, water is the dominant component by mass. In humans, it makes up about 60% of total body weight, and in some simple organisms, it can reach more than 90%. Without it, life as we know it could not exist. The reason water plays such a central role in biochemistry lies in its unique properties, properties that arise from the way its atoms are arranged and how they interact with each other. A water molecule is made of 2 hydrogen atoms and 1 oxygen atom, bonded together in a bent shape. Oxygen holds onto electrons more tightly than hydrogen, creating a slight negative charge near the oxygen atom and a slight positive charge near each hydrogen. This uneven distribution of charge makes water a polar molecule. Polarity is the key to many of water's remarkable traits. Because each water molecule has both a positive and a negative end, it can form weak but important connections, called hydrogen bonds, with other water molecules. These bonds are not as strong as covalent bonds, but in large numbers they create a delicate network that holds water together. It is this network that gives water its unusual stability and its ability to influence life's chemistry in so many ways. One of water's most important abilities is to act as a universal solvent. Many substances dissolve easily in water because its polar molecules can surround and separate ions and polar molecules, breaking them apart into solution. In living systems, this means that salts, sugars, amino acids, and many other molecules essential for life can move freely through the watery environment of a cell. This mobility allows for rapid chemical interactions and efficient transport of nutrients and waste. Water also plays a crucial role in regulating temperature. It has a high specific heat capacity, which means it can absorb or release large amounts of heat with only small changes in its own temperature. This property helps stabilize the internal temperature of organisms, protecting delicate biochemical processes from sudden swings in heat or cold. On a planetary scale, it moderates Earth's climate, absorbing heat in the summer and releasing it in the winter, smoothing out extremes. Another unusual feature of water is the way it behaves when it freezes. Most substances become denser as they solidify, but water is different. As ice forms, hydrogen bonds lock water molecules into a crystalline structure that takes up more space than liquid water. This makes ice less dense, so it floats. This seemingly simple fact has enormous consequences for life. Floating ice insulates the water beneath it, preventing lakes, rivers, and oceans from freezing solid in cold climates and preserving aquatic life through the winter. In cells, water is not only a solvent but also an active participant in many biochemical reactions. In hydrolysis reactions, water molecules are used to break chemical bonds, splitting larger molecules into smaller ones. In condensation reactions, also called dehydration synthesis, water is formed as a byproduct when small molecules join together to form larger structures like proteins and nucleic acids. Water is thus both a medium for chemical change and a material that participates in it. The movement of water across cell membranes is also essential for life. This movement, called osmosis, depends on differences in solute concentration inside and outside the cell. Water naturally flows toward the side with more dissolved substances, seeking to balance concentrations. In plants, osmosis helps maintain the pressure that keeps stems and leaves upright. In animals, it helps regulate the balance of fluids between cells and their surroundings. Water's role in shaping biological structures is subtle but profound. Proteins, for example, fold into specific three-dimensional shapes, partly because of how their amino acids interact with water. Hydrophilic or water-loving amino acids tend to face outward toward the surrounding water, while hydrophobic or water-fearing amino acids cluster inward, away from water. This interplay between water and protein structure determines how proteins function, how they interact with other molecules, and how they carry out the countless reactions needed for life. Even at the level of genetic material, water has its influence. DNA, with its iconic double helix, is stabilized in part by interactions with surrounding water molecules. The polar phosphate groups in DNA's backbone readily interact with water, helping to keep the molecule properly shaped and protected. We might think of water as silent and passive, But in truth, it is constantly in motion, constantly forming and breaking hydrogen bonds, constantly shaping the environment in which life's molecules operate. It flows through the veins of plants, carrying minerals from roots to leaves. It travels through our bloodstream, delivering oxygen and nutrients to cells. It seeps into the smallest spaces between molecules, influencing their shape, movement, and interactions. And beyond its chemical and physical roles, water connects all life through the planet's great cycles. It evaporates from oceans and lakes, travels as vapor through the air, condenses into clouds, and falls again as rain or snow. It carves rivers, shapes landscapes, and sustains ecosystems. The water in your body now may once have been part of a glacier, a tropical storm, or a prehistoric sea. In this way, water is not just a participant in the chemistry of life. It is a thread that runs through all living history, linking the earliest cells to the world we know today. Every drop carries with it the potential for life's chemistry to unfold, and every living thing carries within it the quiet memory of water's role in its existence. As we move forward in our exploration, we will look at how the elements we have met so far, bound together in countless arrangements, form the four great families of biological macromolecules. Each family has its own structure, its own purpose, and its own place in the great network of biochemistry. And in every one of them, water will be there, shaping, supporting, and sustaining the molecular dance of life. 6. The language of molecules. Every living thing is made of molecules, countless, intricate arrangements of atoms, bound together in patterns that nature has been writing for billions of years. These molecules are the words and sentences in life's chemical language. Just as a language has grammar and structure, the molecular world has rules, rules that govern how atoms join, how they hold together, and how they separate. Understanding this language is essential to understanding biochemistry, for it is the means by which life communicates with itself at the most fundamental level. At the foundation of this language are the atoms themselves. Each atom is like a letter in the chemical alphabet, with its own distinct properties. Carbon, oxygen, hydrogen, nitrogen, phosphorus, and sulfur are the main letters in life's script, though others appear in supporting roles. Just as letters can be combined to make countless words, atoms can be arranged into an infinite variety of molecules. The way atoms connect is determined by chemical bonds. The strongest of these are covalent bonds, bonds in which two atoms share one or more pairs of electrons. This sharing allows each atom to fill its outer electron shell, reaching a stable configuration. In biological systems, covalent bonds are the backbone of molecules. They hold the chains of carbon atoms in sugars, the peptide links in proteins, and the nucleotide connections in DNA. Yet covalent bonds are not all alike. Some are nonpolar, where the electrons are shared equally between the atoms, as in the bond between two carbon atoms. Others are polar, where the electrons are drawn more strongly toward one atom than the other, as in the bond between oxygen and hydrogen in water. This uneven sharing creates slight charges on the atoms, giving polar molecules unique abilities to interact with other charged or polar substances. Beyond covalent bonds, there are weaker, but equally important, connections. Hydrogen bonds, for example, arise when a hydrogen atom covalently bonded to an electronegative atom, like oxygen or nitrogen, is attracted to another electronegative atom nearby. Individually, these bonds are fragile and easily broken, but in large numbers, they create powerful effects, shaping the structure of DNA, holding proteins in their folded forms, and giving water its remarkable properties. Another kind of connection is the ionic bond. Here, electrons are transferred rather than shared. One atom gives up an electron and becomes positively charged, Another gains that electron and becomes negatively charged. These opposite charges draw the atoms together. In biological systems, ionic bonds often form between charged side chains of amino acids or between minerals like sodium and chloride, giving rise to table salt. There are also van der Waals forces, faint, temporary attractions that occur when electrons in a molecule momentarily shift to one side, creating a tiny dipole. These forces are weaker than hydrogen bonds, but play a role in how molecules fit together, especially in large biological structures. Together, these bonds and forces form the grammar of life's chemistry. They determine the shapes of molecules, how they interact, and how they move through the watery world inside cells. Shape, in turn, is deeply tied to function. A molecule's ability to bind another molecule, to catalyze a reaction, or to store energy, all depend on the exact arrangement of its atoms in three-dimensional space. Consider proteins, the versatile workhorses of life. Their function depends on folding into precise shapes, and this folding is guided by a network of bonds, covalent peptide bonds linking amino acids, hydrogen bonds stabilizing coils and sheets, ionic bonds between charged groups, and van der Waals forces drawing nonpolar regions together. Even a small change in these bonds can alter a protein's shape, and with it, its ability to function. DNA, too, speaks this molecular language. Its famous double helix is stabilized by hydrogen bonds between complementary base pairs, adenine with thymine, guanine with cytosine, and by stacking interactions between the flat, ring-shaped bases. The specific pairing rules are a form of molecular syntax, ensuring that genetic information can be copied accurately and passed on to the next generation. In biochemistry, the language of molecules is not static. Bonds are constantly forming and breaking, sentences constantly rewritten. Metabolism, the sum of all chemical reactions in a cell, is essentially a conversation in this language. Enzymes act as skilled interpreters, guiding reactions along preferred paths, lowering the energy needed to break bonds, and speeding the assembly of new ones. Even communication between cells is conducted in molecular terms. Hormones, neurotransmitters, and signaling molecules fit into specific receptor proteins the way a key fits into a lock. The shapes must match precisely for the signal to be recognized. This specificity is why a hormone that regulates growth does not accidentally trigger a muscle contraction, and why a neurotransmitter in the brain cannot be mistaken for a digestive enzyme in the stomach. The precision of this language is remarkable. A single misplaced bond, a single atom out of place, can change the entire meaning of a molecular message. Sometimes the result is harmless. Other times it can alter a protein so severely that it no longer functions, leading to disease. In this way, the fidelity of molecular language is as important to life as the fidelity of spoken or written language is to human communication. And yet, despite this precision, the language of molecules is flexible and adaptable. Evolution has shaped it to work in many environments, from the near-freezing waters of polar seas to the boiling depths of hydrothermal vents. Organisms have evolved molecules whose bonds and structures can withstand extremes, adjusting their grammar to the conditions of their world. The more we learn this language, the more we can read the story of life written at the molecular level. We can see how the same basic bonds are used again and again to build vastly different structures. We can see how subtle variations in shape and charge give molecules their unique personalities. We can see, too, how all living things are fluent in the same chemical tongue, sharing the same rules no matter how different they appear on the surface. In the chapters ahead, we will begin to explore the vocabulary of this language in detail, the great families of biological molecules. We will see how carbohydrates store energy and provide structure, how lipids form membranes and store fuel, how proteins act as catalysts and builders, and how nucleic acids store the instructions for life itself. Each of these families uses the same basic chemical rules, but each tells a different part of the story of life. And through it all, the bonds we have met here will be present, the covalent connections holding backbones together, the hydrogen bonds lending shape and stability, the ionic interactions guiding assembly, and the van der Waals forces adding subtle touches of precision. Life speaks in this molecular language at every moment, and as we learn to read it, we come closer to understanding the quiet, intricate conversations that have been taking place in living cells for billions of years. 7. Carbohydrates, energy, and structure. In the grand library of life's molecules, carbohydrates are among the most familiar volumes. They are the sweet, sustaining energy bearers of the living world, but they are also far more than simple fuel. They provide structure, support, and sometimes even signals for communication between cells. Their versatility comes from their ability to form a remarkable variety of shapes and sizes, from the simplest sugar molecule to the most intricate plant fiber. At their core, carbohydrates are composed of just 3 elements: carbon, hydrogen, and oxygen. The ratio of hydrogen to oxygen in these molecules is typically the same as in water, 2 hydrogens for every oxygen, giving them the name carbohydrate, literally meaning carbon plus water. This simple composition hides a vast diversity of forms. The simplest carbohydrates are the monosaccharide single sugar units. The most familiar of these is glucose, the primary fuel for most cells. Glucose is the currency of energy transactions in the body. Cells take it in, break it down, and release its stored chemical energy to power everything from muscle contractions to nerve signals. Other monosaccharides, such as fructose in fruits and galactose in milk, play their own roles, but all share a similar backbone of carbon atoms adorned with hydroxyl OH groups. These simple sugars can join together to form disaccharides, 2-unit sugars linked by covalent bonds. Sucrose, or table sugar, is a disaccharide made of glucose and fructose. Lactose, the sugar found in milk, combines glucose and galactose. Maltose, formed during the breakdown of starch, is a pair of glucose molecules. Disaccharides are often used for transport or storage, easily split back into monosaccharides when needed. When many sugar units are linked together, the result is a polysaccharide. These long chains can be straight or branched, compact or sprawling. Their structure determines their function. Starch, for example, is the main storage form of carbohydrate in plants. It is made of glucose units linked in a way that allows the chain to coil into a dense form, ideal for storing large amounts of energy in a small space. Animals store carbohydrate as glycogen, a more highly branched version of starch, making it easier to quickly release glucose when energy demand suddenly increases. Polysaccharides are not just for storing energy. They also serve as strong building materials. Cellulose, the most abundant organic compound on Earth, is a polysaccharide composed of glucose molecules linked in a way that creates straight, rigid chains. These chains bundle together into fibers that give plant cell walls their strength. Though cellulose is made from the same glucose units as starch, the difference in the type of bond between them means that most animals cannot digest it. In humans, it passes through the digestive system as dietary fiber, aiding digestion, even if it yields no direct energy. In some organisms, polysaccharides serve as protective coatings or as part of the skeleton. Chitin, found in the exoskeletons of insects and the cell walls of fungi, is a polysaccharide similar to cellulose but with nitrogen-containing groups attached. This modification makes it both strong and flexible, ideal for forming protective shells. The ability of carbohydrates to take on so many roles comes from their chemical versatility. Their many hydroxyl groups make them polar and soluble in water, allowing them to dissolve easily for transport within living systems. At the same time, they can form countless branching patterns, allowing for molecules that are small and mobile, or large and rigid. In living cells, Carbohydrates are not always isolated molecules. Often, they are attached to proteins or lipids, forming glycoproteins and glycolipids. These combinations are found on the outer surfaces of cell membranes, where they act as identity markers and communication tools. They help cells recognize each other, distinguish friend from foe, and coordinate activities. In the immune system, for instance, specific carbohydrate patterns on the surface of a pathogen can trigger a defensive response. Carbohydrates also play a role in structural signaling. In developing organisms, complex carbohydrate patterns help guide cells to their correct positions, ensuring that tissues form in the right places. In this way, they act like a molecular map, directing the choreography of growth and development. Energy-wise, carbohydrates are relatively easy for cells to break down. Through processes such as glycolysis and cellular respiration, the bonds in glucose are systematically broken, releasing energy that is stored in molecules of ATP. This ATP can then be used immediately for work within the cell. Because they are so readily available and efficient to use, carbohydrates are often the first choice for quick energy in both plants and animals. Yet, for all their importance, carbohydrates are only one part of the larger molecular picture. They work alongside lipids, proteins, and nucleic acids, each with their own specialties, to keep life running smoothly. Carbohydrates provide the ready energy and, in some cases, the sturdy scaffolding upon which cells and organisms are built. Without them, the very architecture of plants, the quick responses of animals, and the communication between cells would be far less efficient. In the world of biochemistry, carbohydrates remind us of a simple truth. Even the most basic building blocks can form astonishingly complex and varied structures. From the sweetness of fruit to the towering strength of a tree trunk, they quietly shape both the softness and the solidity of life. As we move forward, we will turn to the second great family of biological molecules, lipids. Unlike carbohydrates, lipids are not primarily water-loving. They are the molecules that form life's boundaries, store energy in dense reserves, and carry out many subtle, powerful roles in the chemistry of living things. 8. Lipids, energy storage, and membranes. If carbohydrates are the quick, ready fuel of life, lipids are its deep reserves, concentrated stores of energy that can sustain an organism through long stretches without food. But lipids are not only about storage. They are also the architects of life's boundaries, forming the thin, flexible membranes that separate a cell from the world around it and divide its interior into specialized compartments. Without lipids, there would be no true separation between inside and outside, no protected spaces for life's delicate chemistry to take place. Lipids are a diverse family of molecules, but they share one defining trait. They are largely hydrophobic. They do not mix well with water. This property comes from their structure. While carbohydrates and proteins have many polar groups that readily form hydrogen bonds with water, lipids are dominated by long chains or rings of carbon and hydrogen atoms. These nonpolar regions are uncharged and cannot easily interact with water molecules. In the simplest terms, lipids can be grouped into three main categories: fats and oils, phospholipids, and steroids. Each plays a distinct role in the biochemistry of life. Fats and oils are primarily energy storage molecules. They are made of a glycerol backbone attached to three fatty acid chains, forming a structure called a triglyceride. The fatty acids themselves are long hydrocarbon chains with a carboxyl group at one end. These chains can vary in length and in the number of double bonds they contain. Saturated fatty acids have no double bonds, meaning their carbon atoms are fully saturated with hydrogen atoms. They tend to pack closely together, making fats solid at room temperature. Unsaturated fatty acids have one or more double bonds, introducing bends in the chain that prevent tight packing and make oils liquid at room temperature. Because lipids are hydrophobic, they can store energy in a dense, compact form without drawing in water. This makes them far more efficient for long-term storage than carbohydrates. A gram of fat stores more than twice the energy of a gram of carbohydrate. For animals, this high energy density makes lipids ideal for enduring long migrations, hibernation, or other periods when food is scarce. Phospholipids, by contrast, are the master builders of biological membranes. Each phospholipid has a glycerol backbone and two fatty acid tails, but the third position on the glycerol is occupied by a phosphate group attached to a small polar head group. This gives phospholipids a dual nature. Their tails are hydrophobic, while their heads are hydrophilic, water-loving. When phospholipids are placed in water, they spontaneously arrange themselves so that their hydrophobic tails are shielded from the water, while their hydrophilic heads face outward toward it. In living cells, this results in the formation of a phospholipid bilayer, two layers of phospholipids, with tails facing inward and heads facing outward. This bilayer forms the basic structure of cell membranes. Cell membranes are not rigid barriers. They are flexible, dynamic structures. Proteins float within the bilayer, acting as channels, receptors, and enzymes. Sugars attach to some lipids and proteins, forming glycoproteins and glycolipids that help with cell recognition and communication. Cholesterol molecules nestle among the phospholipids, adjusting membrane fluidity depending on temperature. This arrangement, known as the fluid mosaic model, gives the membrane both stability and adaptability. Steroids form another important class of lipids. Unlike fats and phospholipids, steroids have a structure based on 4 interconnected carbon rings. Cholesterol is the most familiar steroid. It plays multiple roles in the body. It is a component of cell membranes, helping to maintain fluidity, and it is also the precursor from which the body synthesizes steroid hormones, such as estrogen, testosterone, and cortisol. These hormones act as powerful signaling molecules, influencing growth, metabolism, stress responses, and reproductive processes. Not all lipids fit neatly into these categories. Waxes, for example, are long-chain fatty acids linked to long-chain alcohols, forming waterproof coatings on leaves, fruits, and animal fur. Lipid-derived molecules like prostaglandins function as local signaling messengers, involved in processes such as inflammation and blood flow regulation. Beyond their structural and storage roles, lipids have a deep influence on the functioning of life. In nerve cells, specialized lipids form myelin, an insulating sheath that allows electrical impulses to travel rapidly along nerve fibers. In plants, pigments such as carotenoids, which are lipid-soluble, capture light energy for photosynthesis and protect against damage from excessive sunlight. In the skin, lipid layers help prevent water loss and block the entry of harmful substances. Because lipids are hydrophobic, they require special handling for transport in the watery environment of the body. In animals, fats are packaged into structures called lipoproteins, with a surface of phospholipids and proteins surrounding a core of triglycerides and cholesterol. These packages travel through the bloodstream to deliver lipids to cells that need them. Lipids are also deeply tied to health. Their balance in the body matters. Too few lipids, and the body cannot maintain its membranes, store enough energy, or produce hormones. Too many, and they may accumulate in blood vessels, leading to health problems. The composition of lipids in membranes can affect how cells respond to signals, how well they resist temperature changes, and how susceptible they are to damage. In the quiet world inside every cell, lipids work alongside proteins, carbohydrates, and nucleic acids to keep life functioning. They do not shout their importance, but without them, there could be no distinct cell boundaries, no efficient long-term energy storage, and no many-layered system of chemical signals guiding growth, repair, and adaptation. From the soft fat reserves that sustain a migrating bird to the shimmering surface of a soap, bubble-like membrane surrounding a cell, lipids remind us that life depends as much on its boundaries as on what lies within them. They shape the spaces in which life's chemistry can unfold, store the energy to keep that chemistry going, and quietly transmit messages that influence the course of an organism's life. As we continue, we turn to the third great family of biological molecules, proteins. These are the master builders and workers of the cell, crafting structures, catalyzing reactions, and responding to the constant flow of life's needs. 9. Proteins, the workhorses of life. If lipids are the architects of life's boundaries and carbohydrates are its ready stores of energy, proteins are the tireless workers within the builders, the movers, the messengers, and the catalysts that make nearly every process in a living organism possible. In the language of biochemistry, proteins are perhaps the most versatile of all molecules, capable of taking on an astonishing variety of forms and functions. Without them, life's chemistry would grind to a halt. Proteins are made from smaller building blocks called amino acids. In living systems, there are 20 different amino acids commonly used to build proteins. Each amino acid shares a common backbone, a central carbon atom bonded to a hydrogen atom, an amino group, and a carboxyl group. The difference lies in the fourth attachment, called the side chain, or R group. The nature of the side chain, whether it is large or small, polar or nonpolar, acidic or basic, gives each amino acid its unique personality. These amino acids link together in long chains through covalent peptide bonds. A peptide bond forms when the carboxyl group of one amino acid reacts with the amino group of another, releasing a molecule of water in the process. As more amino acids join, the growing chain is called a polypeptide. When the chain folds into its functional shape, it becomes a protein. The sequence of amino acids in a protein is its primary structure, the fundamental blueprint that determines all higher levels of structure. The chain does not remain as a simple line. Chemical interactions cause it to fold, twist, and coil. This folding happens in distinct stages. The secondary structure is formed through regular patterns like the alpha helix, a spiral shape stabilized by hydrogen bonds, and the beta sheet, a flat, pleated arrangement also held by hydrogen bonds. These secondary structures fold further into a three-dimensional form called the tertiary structure, shaped by a combination of forces, hydrogen bonds, ionic interactions, hydrophobic clustering, and even covalent disulfide bonds between sulfur-containing amino acids. The final shape of a protein is intimately tied to its function, like the shape of a key to the lock it fits. Some proteins consist of more than one polypeptide chain. These chains come together in a specific arrangement, forming the quaternary structure. Hemoglobin, the protein that carries oxygen in red blood cells, is a classic example. It consists of 4 polypeptide subunits working together as one functional unit. The range of tasks that proteins perform is extraordinary. Many are enzymes catalysts that speed up chemical reactions without being consumed. Enzymes lower the activation energy needed for a reaction to occur, allowing life's chemistry to proceed at the temperatures and conditions found inside cells. Without enzymes, most biochemical reactions would be far too slow to sustain life. Other proteins serve as structural components. Collagen, for example, is a fibrous protein that gives strength and elasticity to connective tissues such as tendons and skin. Keratin forms the structural basis of hair, nails, feathers, and horns. In cells, cytoskeletal proteins like actin and tubulin create scaffolds that give the cell its shape and allow it to move. Proteins also play essential roles in transport. Hemoglobin carries oxygen from the lungs to tissues. Membrane transport proteins form channels or pumps that move ions and molecules in and out of cells, maintaining the delicate balance of the cell's internal environment. Some proteins act as messengers, transmitting information within and between cells. Hormones like insulin are proteins that signal cells to adjust their metabolism, while receptor proteins embedded in cell membranes detect chemical signals from the outside world. Antibodies, a special class of proteins in the immune system, recognize and bind to foreign molecules, marking them for destruction and helping to protect the organism from harm. Motor proteins are another remarkable group. Myosin in muscle cells, for instance, converts chemical energy from ATP into mechanical force, allowing muscles to contract. Kinesin and dynein move along microtubule tracks within cells, carrying cargo such as vesicles and organelles to their destinations. The flexibility of proteins comes from the diversity of amino acids and the precision of folding. Yet this very complexity makes them sensitive to changes. If a protein is exposed to high temperatures, extreme pH, or certain chemicals, its structure can unravel in a process called denaturation. Once unfolded, the protein often loses its function, and in living organisms, this can have serious consequences. Cells take great care to ensure proteins fold correctly. Specialized helper molecules called chaperone proteins assist in the folding process, preventing incorrect interactions and guiding the polypeptide into its proper shape. Misfolded proteins can lead to diseases such as Alzheimer's, Parkinson's, and cystic fibrosis, reminding us how much depends on the delicate architecture of these molecules. Proteins are also dynamic. Many change shape in response to binding other molecules, switching between active and inactive forms. This ability to change conformation allows them to act as molecular switches, turning processes on or off as needed. In signaling pathways, a protein's shape change might trigger a cascade of reactions that spreads through the cell like a quiet but decisive ripple. In diet and nutrition, proteins are essential not only because they can be used for energy if necessary, but because they provide the amino acids the body cannot make on its own. These essential amino acids must come from food, and once absorbed, they are reused to build the body's own proteins. The constant turnover of proteins, their breakdown and resynthesis, is a sign of life's ongoing renewal at the molecular level. In the quiet interior of every cell, proteins are at work. Some are holding the cell's shape steady, others are shuttling materials from place to place, others are building, repairing, signaling, or defending. Together, they are the workforce of life, present in every corner of the living world, endlessly adaptable and endlessly necessary. As we continue our journey, we turn to the fourth great family of biological molecules, nucleic acids. These are the molecules of memory and instruction, holding the information that tells proteins how to form and guiding the growth, function, and reproduction of every living thing. 10. Nucleic acids, the code of life. Proteins may be the workers of life, building and maintaining every structure, catalyzing every reaction, but they do not design themselves. Their precise shapes and sequences must be specified in advance. This design is stored in molecules that act as life's memory molecules that preserve instructions generation after generation and guide the construction of every protein in every cell. These are the nucleic acids, the molecules of genetic information. There are two main types of nucleic acids in living systems: DNA, or deoxyribonucleic acid, and RNA, or ribonucleic acid. Together they hold and transmit the instructions for life's processes, but each plays its own role in the flow of genetic information. DNA is the primary storehouse of genetic information. In humans and most other organisms, it resides in the cell nucleus, coiled and packaged into chromosomes. A molecule of DNA is like an extraordinarily long sentence written in a simple alphabet of just 4 letters. Each letter is a nucleotide, made of 3 components: a sugar molecule, a phosphate group, and a nitrogen-containing base. The sugar in DNA is deoxyribose, and the phosphate group links the sugars into a repeating backbone. The bases are what carry the actual information. There are 4 kinds: adenine, A, thymine, T, cytosine, C, and guanine, G. These bases do not arrange themselves randomly. They pair in a consistent way: adenine always with thymine, cytosine always with guanine, through hydrogen bonds. These pairings are the rungs of DNA's famous double helix, a twisted ladder of information. The order of bases along a DNA strand is a code. Groups of bases form genes, and each gene carries the instructions for building a specific protein. The sequence of bases determines the sequence of amino acids in that protein, which in turn determines how it folds and functions. A single change in this sequence, one letter swapped for another, can sometimes alter a protein's function dramatically, underscoring the precision of this molecular code. RNA is closely related to DNA, but has important differences. Its sugar is ribose rather than deoxyribose, and instead of thymine, RNA uses uracil, U, as one of its 4 bases. RNA is usually single-stranded, allowing it to fold into a variety of shapes for different tasks. While DNA serves as a stable archive, RNA is more like a working copy or messenger, carrying genetic information from DNA to the cellular machinery that builds proteins. The central process linking DNA, RNA, and proteins is called the central dogma of molecular biology. DNA is transcribed into RNA, and RNA is translated into protein. Transcription begins when a section of DNA unwinds, exposing the base sequence of a gene. An enzyme called RNA polymerase reads one strand of the DNA and builds a complementary RNA strand. This messenger, RNA or mRNA, carries the genetic instructions out of the nucleus to the ribosomes, the molecular machines that make proteins. Translation occurs in the ribosomes. Here, the sequence of bases in mRNA is read in groups of 3, called codons. Each codon specifies a particular amino acid. Transfer RNA, or tRNA, molecules bring the correct amino acids to the ribosome, where they are linked together in the order dictated by the mRNA. This chain of amino acids will fold into a protein with a specific function. Not all RNA is a messenger. Some RNAs help build the ribosome itself, ribosomal RNA, or rRNA, while others assist in protein synthesis, tRNA. Still others act as regulators, controlling when and how much of a protein is made, or even silencing certain genes altogether. These regulatory RNAs are vital to fine-tuning the expression of the genetic code. The beauty of nucleic acids lies in their dual nature. They are both the blueprint for life and, through their interactions with proteins and enzymes, active participants in carrying out that blueprint. The DNA you carry in your cells today is the product of countless generations before you, stretching back to the first living cells on Earth. Every breath you take, every heartbeat, every thought is sustained by proteins built according to instructions written long ago and faithfully copied ever since. Replication is the process by which DNA copies itself, so that genetic information can be passed on to new cells and to offspring. During replication, the 2 strands of the DNA double helix separate, and each serves as a template for building a new complementary strand. Enzymes called DNA polymerases add nucleotides one by one, matching them to their partners on the template strand A with T, C with G. The result is 2 identical DNA molecules, each with one old strand and one new strand. This method, called semi-conservative replication, ensures remarkable fidelity in preserving the code. Yet errors can occur, and these mutations are a source of both risk and possibility. Some mutations are harmful, disrupting essential proteins. Others are neutral, having no noticeable effect. A few may even be beneficial, providing new traits that help an organism adapt to its environment. Over long spans of time, such changes are the raw material of evolution. Nucleic acids also carry the marks of life's shared history. The genetic code is nearly universal, used by almost all known organisms. The codon that specifies the amino acid methionine in a bacterium is the same codon that does so in a human. This universality is powerful evidence that all life on Earth shares a common origin. In the context of biochemistry, nucleic acids are more than abstract information. They are physical molecules with shape, charge, and chemical properties. DNA's double helix is stabilized by hydrogen bonds between bases and by stacking interactions between the flat, ring-shaped bases themselves. RNA's ability to fold into complex shapes allows it to act like an enzyme in some cases, catalyzing reactions, a reminder that the boundary between information and action is not always sharp. Life depends on the precise interplay between nucleic acids and the other great families of molecules. Without proteins, the instructions in DNA could never be carried out. Without lipids to form membranes, nucleic acids could not be kept safe within cells. Without carbohydrates, cells could not generate the energy needed to copy DNA or build RNA. And yet, it is the nucleic acids that hold the continuity of life's story. They are the reason a child inherits traits from a parent, the reason a tree can grow from a seed, the reason a bacterium can divide and produce an identical twin. They are the keepers of life's memory and the architects of its future. As we continue our journey, we will turn our attention to one of the most remarkable kinds of proteins, enzymes, which serve as the catalysts for nearly every chemical transformation in living systems. They are the tools that bring the instructions written in nucleic acids to life, speeding reactions that would otherwise move too slowly to sustain existence. 11. Enzymes, nature's gentle catalysts. Every moment within a living cell, countless chemical reactions are taking place. Molecules are being broken down, built up, rearranged, and transformed. These reactions form the essence of life's chemistry, yet under normal conditions, many of them would occur far too slowly to sustain life. The gap between what is possible in theory and what happens in the living world is bridged by a remarkable class of proteins, enzymes. Enzymes are catalyst substances that increase the speed of a chemical reaction without being consumed or permanently changed by it. In this way, they can be used again and again, quietly guiding reactions along their paths. Without enzymes, even the most vital processes, such as the breakdown of glucose for energy or the copying of DNA, would proceed at a pace so slow that life as we know it could not exist. What makes enzymes so powerful is their ability to lower the activation energy of a reaction. Activation energy is the initial input of energy needed to get a reaction started, the small push required before a boulder begins to roll downhill. By lowering this barrier, enzymes make it possible for reactions to proceed rapidly, even at the gentle temperatures and pressures found in living cells. Enzymes achieve this with extraordinary precision. Each enzyme is shaped in a way that allows it to bind to specific molecules, called substrates. The region of the enzyme where the substrate binds is known as the active site. This site is not just a passive pocket. It is a carefully arranged landscape of atoms, designed to hold the substrate in exactly the right position to encourage the reaction to occur. When a substrate enters the active site, the enzyme may change shape slightly in what is known as the induced fit model. This subtle shift helps bring chemical groups into alignment, weakening certain bonds or positioning molecules so they can react more easily. Once the reaction has taken place, the enzyme releases the products and is free to catalyze the process again with a new substrate. The specificity of enzymes is one of their most remarkable qualities. Most enzymes act on only one type of substrate, or on a very small group of related molecules. This ensures that the cell's chemistry remains orderly, with each enzyme performing its task without interfering with others. The names of enzymes often reflect this specificity, typically ending in "ase" and indicating the reaction they catalyze. For example, lactase breaks down lactose, and DNA polymerase builds new strands of DNA. Enzymes are involved in every major pathway of metabolism. In the digestion of food, enzymes such as amylase, protease, and lipase break down carbohydrates, proteins, and fats into smaller units that cells can absorb. In cellular respiration, enzymes guide the step-by-step release of energy from glucose, capturing it in the form of ATP. In DNA replication and repair, specialized enzymes carefully copy genetic material and correct mistakes, preserving the integrity of the code. Some enzymes require the help of additional molecules to function. These helpers are known as cofactors. A cofactor can be a metal ion, such as magnesium or zinc, or an organic molecule called a coenzyme, often derived from vitamins. These cofactors may assist in stabilizing the structure of the enzyme or participate directly in the reaction, carrying chemical groups or electrons from one molecule to another. The activity of enzymes is not fixed. It is finely regulated by the cell, temperature, pH, and the concentration of substrates all influence how efficiently an enzyme works. Enzymes typically function best within a narrow range of conditions, and deviations can slow their activity or even cause them to unfold and lose their shape, a process known as denaturation. Cells can also regulate enzymes more directly. Inhibitors are molecules that slow or stop enzyme activity. Some inhibitors bind to the active site, blocking the substrate from entering. These are called competitive inhibitors. Others bind to a different part of the enzyme, altering its shape so that the active site no longer functions properly. These are non-competitive inhibitors. Regulation of enzymes ensures that reactions happen only when and where they are needed, conserving energy and resources. Conversely, activators can enhance enzyme activity, often by helping the enzyme maintain its active shape, or by increasing the likelihood that a substrate will bind. Through this interplay of inhibition and activation, cells can adjust their metabolism moment by moment, responding to changes in their environment. One of the most elegant examples of enzyme control is feedback inhibition. In this process, the end product of a metabolic pathway inhibits an enzyme that acts early in the pathway. When the product accumulates, it slows its own production, preventing waste. When levels drop, the inhibition is lifted and production resumes. This gentle, self-regulating cycle helps maintain balance within the cell. Enzymes can also display an impressive ability to work in sequence. In metabolic pathways, the product of one enzyme often becomes the substrate for the next. Like an assembly line, each enzyme performs a specific step, gradually transforming a starting molecule into a final product. This ordered progression ensures efficiency and coordination in the cell's complex chemical network. Some enzymes are so efficient that they approach the theoretical maximum speed at which a substrate molecule can diffuse into the active site. These catalytically perfect enzymes seem to operate at the limits of what is physically possible, turning over substrates as fast as they encounter them. Others work more slowly, but with exquisite precision, catalyzing complex transformations that would be impossible without them. Beyond their role in natural life processes, enzymes have become indispensable in medicine, industry, and research. In medicine, enzymes are used in diagnostic tests, in treatments for certain diseases, and in the development of new drugs. In industry, they are employed in the production of food, beverages, textiles, and biofuels, often replacing harsher chemical methods with cleaner, more sustainable processes. In the laboratory, enzymes are essential tools for studying and manipulating DNA, proteins, and other molecules. Yet for all their versatility and power, enzymes remain humble in their work. They do not drive reactions with brute force, but with subtle guidance, creating a favorable environment for chemistry to happen naturally. In the stillness of a cell, they go about their tasks with quiet efficiency, ensuring that life's chemistry runs smoothly and continuously. As we move forward in our exploration, we will step back and look at the broader picture of how enzymes and other molecules manage the energy of life. In the next part, we will turn to the concept of energy within cells, how it is captured, stored, and used to sustain the constant motion and transformation of living systems. 12. Energy in the cell. Life is movement. Even when we are still, countless processes unfold inside us. Molecules are transported, signals are sent, proteins are built, waste is removed. Each of these activities requires energy, a constant, reliable flow of it. Without this energy, the machinery of life would stop. The cell would fall silent. Energy is the capacity to do work, to cause change. In living systems, work means everything from contracting a muscle to moving a molecule across a membrane, from building a complex protein to transmitting a nerve impulse. The source of this energy is not magic, but the careful transformation of matter and the breaking and making of chemical bonds. The primary energy currency of the cell is a small molecule called adenosine triphosphate, or ATP. ATP is to the cell what money is to an economy, a convenient, portable form of value that can be spent on any number of activities. Structurally, ATP is composed of three parts: the nitrogen-containing base adenine, the sugar ribose, and a chain of three phosphate groups. It is in these phosphate groups, and the bonds between them, that ATP's energy is stored. The last phosphate bond, in particular, is high in potential energy. When ATP is broken down to adenosine diphosphate, ADP, and a free phosphate group, this bond is broken, and energy is released. The cell captures this energy and uses it to power countless reactions. This is not an uncontrolled burst of energy, but a measured release, carefully matched to the needs of the process at hand. The constant regeneration of ATP is vital. A human body contains only a small amount of ATP, at any given moment enough for just a few seconds of intense activity, yet we use our body weight in ATP many times over each day. Cells recycle ATP continuously, replenishing it from ADP and phosphate through processes like cellular respiration and, in plants, photosynthesis. Cellular respiration is the main pathway for generating ATP in most organisms. It begins with glycolysis, a sequence of reactions that breaks down glucose into smaller molecules, releasing a modest amount of energy and producing a few molecules of ATP. The products of glycolysis enter the mitochondria, the cell's energy powerhouses, where the Krebs cycle and the electron transport chain extract far more energy, capturing it in the form of additional ATP. Oxygen plays a critical role here, acting as the final acceptor of electrons in the chain, allowing the process to continue. In environments where oxygen is scarce, cells can still produce ATP through anaerobic pathways, such as fermentation. These processes are less efficient, yielding fewer ATP molecules per glucose, but they can sustain life when oxygen is not available. Many microorganisms rely on such pathways entirely, and even human muscle cells turn to them temporarily during intense exertion. ATP is not the only form of energy storage in cells. Energy can also be held in other molecules, carbohydrates like glycogen, fats stored in lipid droplets, and even in high-energy electrons carried by coenzymes such as NADH and FADH2s. These molecules act as reserves, ready to be converted into ATP when the cell's immediate needs rise. The management of energy in a cell is a delicate balance. Too little ATP in essential processes slow or stop. Too much, and the cell risks wasting valuable resources. Cells respond to energy demand with fine-tuned control, speeding up ATP production when activity increases, and slowing it when demands fall. This balance is maintained by sensors and regulatory proteins that monitor the cell's energy status and adjust metabolism accordingly. Energy is not only needed for movement and building, it is also required to maintain order. Living systems naturally tend toward disorder, over time, a tendency described by the second law of thermodynamics. Cells must continually use energy to maintain their internal structure, repair damage, and keep conditions within narrow ranges that support life. This constant work is what separates the living from the non-living. The flow of energy through living systems is part of a much larger cycle. Energy enters most ecosystems through sunlight, captured by plants, algae, and certain bacteria in the process of photosynthesis. These organisms convert light energy into chemical energy stored in glucose and other molecules. Animals and other heterotrophs consume these molecules, breaking them down to release their stored energy as ATP. In this way, the energy that begins in the sun flows through the biosphere, sustaining all forms of life. Within the cell, the movement of energy is often coupled. One reaction's release of energy drives another reaction's need for it. This coupling is essential for efficiency. For example, the breakdown of glucose in respiration releases energy, which is immediately used to produce ATP, which in turn fuels processes like active transport across membranes or the synthesis of new molecules. Nothing is wasted. Energy flows from one task to the next, a seamless chain of use and renewal. Even the smallest actions we take, blinking, breathing, thinking, are powered by the quiet work of ATP and the energy transformations it supports. We are, in a sense, walking networks of energy exchange, constantly drawing in, converting, using, and renewing the energy we need to stay alive. As we continue our journey, we will look more closely at how all of these energy-producing and energy-using reactions come together in the great web of metabolism. This is the cell's economy, a vast and interconnected system of chemical pathways that keep life in motion. 13. Metabolism, life's balancing act. In every living cell, there is a constant hum of activity, a ceaseless weaving of chemical reactions that transform matter and energy into the materials and processes of life. This vast and intricate network of reactions is called metabolism. It is the grand choreography of biochemistry, the full spectrum of the cell's chemical life. Metabolism is not one single reaction, but the sum of all the chemical changes that occur within an organism. Some of these reactions break down molecules to release energy. Others use energy to build complex structures from simpler components. These two sides of metabolism, the breaking down and the building up, are known as catabolism and anabolism. Catabolism is the set of pathways that break larger molecules into smaller ones, releasing energy in the process. The digestion of food is the most familiar example. Carbohydrates are broken into simple sugars, proteins into amino acids, fats into fatty acids, and glycerol. These smaller molecules are then further degraded inside cells, often ending in the production of ATP. Catabolic reactions supply the cell with both energy and the raw materials needed for other processes. Anabolism is the opposite. It is the set of pathways that build larger, more complex molecules from smaller ones. When a cell constructs new proteins from amino acids, new DNA strands from nucleotides, or new membranes from fatty acids, it is engaging in anabolism. These processes require an input of energy, often supplied by ATP, to form the chemical bonds that hold the larger molecules together. The balance between catabolism and anabolism is central to life. If catabolism dominates without sufficient anabolism, the organism will gradually deplete its resources, breaking down structures faster than they are rebuilt. If anabolism outweighs catabolism without enough available energy, growth and repair will falter. In healthy living systems, these two forces are in continuous interplay, adapting moment by moment to the needs of the cell and the organism as a whole. Metabolism is organized into pathways series of linked reactions, in which the product of one reaction becomes the substrate for the next. These pathways can be linear, moving step by step toward a single product, or they can branch and interconnect, forming networks with many possible outcomes. The cell's metabolism is not a simple chain but a complex web, with countless points of regulation to direct the flow where it is most needed. One of the most elegant aspects of metabolism is its efficiency. Cells often reuse intermediate molecules that appear in multiple pathways. This sharing of resources minimizes waste and allows the cell to adapt quickly to changes in supply and demand. If glucose is abundant, for example, it can be channeled into energy production through respiration. If energy is plentiful, the same glucose can be diverted into building glycogen for storage. Enzymes are the gatekeepers of metabolic pathways. Each step in a pathway is catalyzed by a specific enzyme, and the activity of these enzymes can be increased, decreased, or halted entirely in response to signals from the cell. This control ensures that resources are not wasted, and that energy flows where it is most needed. Metabolism is also influenced by the availability of raw materials. When certain nutrients are scarce, the cell can slow or stop pathways that depend on them. Conversely, when a nutrient is abundant, the cell can speed up pathways that use it. Hormones play a key role in coordinating metabolism across the body. Insulin, for example, signals cells to take up glucose and store it, while glucagon prompts cells to release glucose from storage when blood sugar is low. Temperature and pH can also affect metabolism, since enzymes work best within certain ranges. Too much heat can cause enzymes to lose their shape. Too little can slow their movement and reduce their efficiency. Similarly, if the internal pH drifts too far from the ideal, enzymes may no longer function properly, disrupting entire pathways. Metabolism is not just about producing energy for immediate needs. It is also about preparing for the future. In times of plenty, organisms store energy in long-term reserves: glycogen in the liver and muscles, fats in adipose tissue. These stores can be drawn upon during fasting, migration, hibernation, or other periods when food is scarce. This ability to store and later release energy is one of life's great survival strategies. Even at rest, metabolism is active. The energy required to maintain the body's basic functions: breathing, circulation, temperature regulation, and the constant repair of tissues is called the basal metabolic rate. This baseline activity sustains life's quiet undercurrent, even when we are asleep or still. On a larger scale, metabolism links the individual to the environment. The molecules we take in as food were once part of other living things: plants, animals, or microorganisms, and the waste products we release return to the environment to be used again. In this way, metabolism connects us to the great cycles of carbon, nitrogen, and other elements that flow through all life on Earth. Metabolism is also adaptable. When conditions change, when we exercise, fast, or encounter stress, the balance of metabolic pathways shifts to meet new demands. Cells can switch fuel sources, relying more on fats when carbohydrates are scarce, or turning to amino acids when both are limited. This flexibility allows life to persist in changing and sometimes challenging environments. The harmony of metabolism comes from its ability to integrate all aspects of cellular activity. Energy, matter, and information flow together, coordinated by enzymes, hormones, and signaling molecules. Each pathway is a thread in a larger fabric, woven so tightly that pulling one can affect many others. In essence, metabolism is life's balancing act, the ongoing negotiation between breaking down and building up, between using and saving, between the needs of the moment and the needs of the future. It is the quiet rhythm that keeps cells alive, bodies functioning, and species enduring. From here, we will explore one of the central catabolic processes in greater detail: cellular respiration. This is the process by which cells harvest energy most efficiently from glucose, capturing it in the form of ATP and fueling the countless reactions that make life possible. 14. Cellular respiration. Every cell in your body requires energy to function, a steady supply of fuel to power the quiet but essential processes of life. This fuel is not used directly in its original form. Instead, it must be converted into the universal energy currency of the cell, ATP. The most efficient and widespread way to produce ATP in living organisms is through a process known as cellular respiration. Cellular respiration is a beautifully orchestrated sequence of biochemical steps that transforms the chemical energy stored in glucose into ATP. Although we often associate it with oxygen, and indeed oxygen is essential for its most efficient form, the process begins in the same way whether or not oxygen is present. The first stage is glycolysis, a 10-step pathway that takes place in the cytoplasm, outside the mitochondria. Here, a single molecule of glucose, a 6-carbon sugar, is gradually broken into 2 molecules of pyruvate, each containing 3 carbons. Along the way, small amounts of energy are captured in the form of ATP and high-energy electrons carried by NADH. Glycolysis does not require oxygen, and for many organisms, especially certain bacteria, it is the sole source of ATP. In more complex organisms, it is just the first chapter in a longer story. When oxygen is available, the pyruvate molecules formed in glycolysis enter the mitochondria, the cell's dedicated energy factories. There, each pyruvate is converted into a 2-carbon molecule called acetyl-CoA, releasing carbon dioxide as a byproduct. This step marks the transition into the second stage of respiration, the citric acid cycle, also known as the Krebs cycle. The citric acid cycle is a circular pathway that systematically removes electrons from the carbon atoms in acetyl-CoA. With each turn of the cycle, more carbon dioxide is released, and more high-energy electron carriers, NADH and FADH2s, are produced. A small amount of ATP is also generated directly in this cycle. The carbon dioxide that we exhale with every breath comes largely from these reactions. The final and most productive stage of cellular respiration is the electron transport chain, located in the inner membrane of the mitochondria. Here, the NADH and FADH2's molecules, generated in the earlier stages, deliver their high-energy electrons to a chain of protein complexes embedded in the membrane. As the electrons pass down the chain, they release energy, which is used to pump protons across the membrane, creating an electrochemical gradient. This gradient represents stored potential energy like water held behind a dam. The protons then flow back across the membrane through a remarkable protein complex called ATP synthase. As they pass through, ATP synthase harnesses the flow to attach phosphate groups to ADP, forming ATP. This process, known as chemiosmosis, produces the vast majority of the ATP generated during cellular respiration. At the very end of the electron transport chain, oxygen serves as the final electron acceptor. It combines with the spent electrons and protons to form water. Without oxygen, the chain would back up, electron flow would stop, and ATP production would quickly falter. This is why oxygen is so vital to aerobic life. It is the molecule that keeps the entire system moving. From one molecule of glucose, aerobic respiration can yield around 30 to 32 molecules of ATP, an extraordinary return compared to the mere 2 ATP produced by glycolysis alone. This efficiency allows complex organisms to sustain demanding processes, from the constant beating of the heart to the firing of neurons in the brain. When oxygen is scarce or absent, cells switch to anaerobic pathways to keep producing ATP. In muscle cells, pyruvate from glycolysis is converted into lactate, allowing glycolysis to continue, but yielding far less ATP overall. In yeast and certain bacteria, pyruvate is instead converted into ethanol and carbon dioxide. These pathways are less efficient, but they are vital survival strategies in oxygen-limited environments. Cellular respiration is more than just a way to make ATP. It is a hub where many metabolic pathways converge. Fats can be broken down into components that feed into the citric acid cycle. Proteins, after their amino groups are removed, can also enter the cycle at various points. In this way, respiration links the metabolism of carbohydrates, fats, and proteins into one integrated system. The mitochondria that carry out respiration are themselves remarkable. They have their own DNA and ribosomes, a legacy of their ancient origin as free-living bacteria that entered into a symbiotic relationship with early eukaryotic cells. Over millions of years, they became permanent residents, passing from one generation to the next, quietly generating the energy needed for life. Every breath you take, every heartbeat, every movement of your muscles depends on this steady transformation of food into ATP. The oxygen you inhale is drawn deep into your lungs, diffuses into your blood, and is carried to every cell, where it slips into the mitochondria to keep the electron transport chain running. The carbon dioxide you exhale is the inevitable byproduct of breaking down the carbon-rich molecules that once held the sun's energy. Cellular respiration is a quiet, continuous cycle, one that sustains life moment by moment. It is not dramatic or sudden, but steady and enduring, a constant undercurrent that powers every other process in the cell. From here, our journey will shift to another process that mirrors respiration in some ways but runs in reverse: photosynthesis. While respiration releases the energy stored in glucose, photosynthesis captures energy from sunlight to build glucose in the first place. Together, these two processes form a great biological loop, connecting the green leaves of plants to the breath of animals in an endless exchange. 15. Photosynthesis, capturing light. Long before animals roamed the earth, before the seas teemed with complex life, a quieter, simpler transformation began to unfold, one that would forever change the planet. Certain early microorganisms developed the ability to capture light from the sun and use it to make food from carbon dioxide and water. This process, photosynthesis, became the foundation of nearly every ecosystem on Earth. It is the source of the oxygen we breathe, the beginning of most food chains, and the means by which sunlight's energy is woven into the molecules of life. Photosynthesis is, in essence, the reverse of cellular respiration. Where respiration breaks down glucose to release energy, photosynthesis uses energy from sunlight to build glucose from smaller molecules. In doing so, it also releases oxygen into the atmosphere, a gift from the green world to all aerobic life. In plants, photosynthesis takes place primarily in the leaves, within specialized cell structures called chloroplasts. Chloroplasts are surrounded by a double membrane and contain stacks of flattened sacs called thylakoids, arranged in groups known as grana. The thylakoid membranes are where the first steps of photosynthesis occur, for they are studded with pigment molecules, the most famous of which is chlorophyll. Chlorophyll is what gives plants their green color, but more importantly, it is a master at absorbing light. It captures energy most efficiently from the blue and red portions of the spectrum, reflecting green light back to our eyes. When a chlorophyll molecule absorbs a photon of light, one of its electrons is excited to a higher energy state. This tiny leap of an electron is the beginning of the chain of events that will ultimately produce sugar and oxygen. Photosynthesis can be thought of in two main stages: the light-dependent reactions and the light-independent reactions, also called the Calvin cycle. The light-dependent reactions occur in the thylakoid membranes. Here, the energy from sunlight is used to split water molecules into oxygen, protons, and electrons. The oxygen is released into the air, a constant, silent flow that sustains the breathing of all animals. The electrons and protons are used to create molecules of ATP and NADPH, two forms of chemical energy that will be used in the next stage. These reactions involve two linked systems known as photosystem II and photosystem I. In photosystem II, light energy drives the splitting of water and the transfer of electrons into an electron transport chain. As electrons move along this chain, their energy is used to pump protons into the thylakoid interior, creating a gradient. Just as in cellular respiration, this gradient drives ATP synthase to produce ATP. The electrons eventually reach photosystem I, where they are energized again by light and used to produce NADPH. The ATP and NADPH produced in the light-dependent reactions are not long-term energy stores. They are immediate forms of energy, ready to be used in the second stage, the Calvin cycle. The Calvin cycle takes place in the stroma, the fluid surrounding the thylakoids inside the chloroplast. Unlike the light-dependent reactions, it does not require light directly, but it depends entirely on the products of the light reactions. Using ATP and NADPH, the Calvin cycle fixes carbon dioxide from the air into organic molecules. Through a series of enzyme-guided steps, carbon dioxide is combined with a 5-carbon sugar called ribulose bisphosphate, RuBP, and gradually transformed into glucose. One of the key enzymes here is Rubisco, often called the most abundant protein on Earth. Its job is to attach carbon dioxide to RuBP, beginning the chain of reactions that leads to sugar production. Although Rubisco works slowly compared to many enzymes, the sheer number of chloroplasts and the vast surface area of green leaves make up for this, allowing plants to draw enormous amounts of carbon from the air over time. The glucose produced by photosynthesis serves many purposes. Plants can use it immediately for energy through respiration, or they can store it as starch for later use. It can also be converted into cellulose to build strong cell walls, or into other organic molecules needed for growth and reproduction. Photosynthesis is not confined to plants alone. Many algae and certain bacteria also perform it, each with variations suited to their environments. Some use pigments other than chlorophyll to capture light, extending the range of colors they can use for energy. In ocean waters, photosynthetic plankton microscopic drifting organisms are responsible for producing a large share of the world's oxygen and play a crucial role in regulating the planet's climate. The impact of photosynthesis reaches far beyond the organisms that perform it. It is the reason our atmosphere contains oxygen, the gas that fuels cellular respiration in animals, fungi, and many bacteria. It is the starting point of the food chain for nearly all life on Earth, capturing the sun's energy and passing it upward through herbivores, carnivores, and decomposers. On a planetary scale, photosynthesis is a stabilizing force. It draws carbon dioxide from the atmosphere, helping to regulate the planet's climate. Over geological time, the oxygen produced by photosynthesis allowed the evolution of more complex life forms, leading eventually to the diversity we see today. It is humbling to realize that every breath we take is linked to this ancient process. The oxygen in your lungs at this moment may have been released by a rainforest tree, a blade of grass, or a tiny plankton cell drifting in the open ocean. The energy in the food you eat began as sunlight, transformed into chemical form by photosynthetic organisms. In the great cycle of life, photosynthesis and respiration are two halves of a whole. Plants and other photosynthesizers capture and store energy in the bonds of glucose, releasing oxygen in the process. Animals and other respiring organisms consume this glucose, using its energy to produce ATP and return carbon dioxide to the air where plants can use it again. From here, our journey will move from the grand scale of planetary energy exchange back to the molecular level of life's instructions. In the next part, we will explore genetic biochemistry, how the code written in DNA is read, translated, and used to guide the building of proteins, completing the connection between energy, structure, and information. 16. Genetic biochemistry. Within every living cell, there is a quiet but constant flow of information, a molecular conversation that connects the stored instructions of life to the active work of building and maintaining the organism. This conversation is the essence of genetic biochemistry, the study of how genetic information is stored, read, and translated into the molecules that carry out life's functions. At the heart of this process is DNA, the molecule we explored earlier as the carrier of genetic information. DNA is more than just a storehouse of instructions. It is a carefully organized library where each gene is a chapter in the story of an organism's life. But unlike a printed book, DNA is not read all at once. Instead, specific genes are selected and transcribed into messages when their instructions are needed. The process begins with transcription. Inside the cell nucleus, the DNA double helix unwinds to expose the sequence of bases along one strand. An enzyme called RNA polymerase binds to a specific starting point on the DNA, guided by signals in the genetic code. Moving along the strand, it reads the sequence of bases and assembles a complementary strand of messenger RNA, mRNA. This mRNA is a working copy of the gene, a portable version of the instructions that can leave the safety of the nucleus. Before it does, in eukaryotic cells, it often undergoes modifications. A protective cap is added to the front. A tail of repeating adenine bases is added to the end, and non-coding sections called introns are removed. These changes help stabilize the message and ensure it is read correctly. Once prepared, the mRNA travels out of the nucleus into the cytoplasm, where it encounters the ribosomes, the molecular machines that build proteins. Ribosomes are themselves made of both protein and ribosomal RNA, rRNA, and they can be thought of as the translators in the genetic conversation. The ribosome reads the mRNA sequence in sets of 3 bases, called codons. Each codon corresponds to a specific amino acid, the building blocks of proteins. Bringing these amino acids to the ribosome are molecules of transfer RNA, tRNA. Each tRNA carries a particular amino acid and has an anticodon, a set of 3 bases that matches a codon on the mRNA. When the anticodon pairs with its matching codon, the ribosome links the amino acid into the growing protein chain. This process, known as translation, continues codon by codon until the ribosome reaches a stop signal in the mRNA. At that point, the completed protein is released, ready to fold into its functional shape and take on its role in the cell. The journey from DNA to protein, from transcription to translation, is often called the central dogma of molecular biology. It is the fundamental flow of information in all known life. DNA makes RNA, and RNA makes protein. Proteins then perform the vast majority of cellular tasks, from catalyzing reactions to forming structures to carrying signals. But genetic biochemistry is not just about the direct creation of proteins. It is also about regulation, controlling when and how much of a protein is made. Cells do not turn on all their genes at once. Instead, they respond to internal signals, such as energy levels or developmental cues, and to external signals, such as temperature changes or the presence of nutrients. Some genes are expressed continuously, producing proteins needed for basic survival. Others are switched on only in response to specific conditions. Regulatory proteins and small RNA molecules can bind to DNA or mRNA, blocking or promoting transcription and translation. This fine-tuning allows cells to conserve resources and adapt to their environment. Errors in the genetic process can have profound effects. A mutation, a change in the DNA sequence, may alter the amino acid sequence of a protein, potentially changing its shape and function. Some mutations are harmless. Others cause disease, and a few may provide beneficial traits that, over generations, contribute to evolution. Genetic biochemistry also extends to the ways DNA itself is maintained and protected. Cells have elaborate repair systems that detect and fix errors in DNA, whether caused by normal wear and tear, environmental factors, or accidental damage. Without these systems, mutations would accumulate quickly, threatening the stability of the genome. In recent decades, our understanding of genetic biochemistry has deepened with the ability to read and edit genetic sequences directly. Techniques such as DNA sequencing have revealed the complete genetic blueprints of many organisms. Tools like CRISPR-Cas9 now allow scientists to make precise changes to DNA, opening possibilities for treating genetic diseases, improving crops, and exploring the functions of specific genes. Yet, despite these technological advances, the essence of genetic biochemistry remains unchanged. It is the careful transfer of information from one molecular form to another, linking the stable memory of DNA to the dynamic work of proteins. This flow of information is what allows a fertilized egg, a single cell, to grow into a complex organism with billions or trillions of specialized cells. It is what allows a plant to respond to sunlight, a bacterium to sense food in its surroundings, and a human body to repair itself after injury. From here, we will explore the next part of our story: how cells use molecular communication to coordinate their activities, not just within a single cell, but across tissues and throughout entire organisms. It is the next layer of life's intricate network, where biochemical messages pass quietly but decisively between different parts of a living system. 17. Molecular communication. Life is not only about the individual work of each molecule inside a cell. It is also about coordination, the ability of molecules to send and receive messages, to act in harmony with one another, and to adjust their activity in response to changing conditions. This is the realm of molecular communication, the subtle language by which cells stay connected to themselves and to the larger systems they belong to. At the most basic level, molecular communication happens within a single cell. Proteins, nucleic acids, ions, and other molecules continually exchange information about the cell's state. Some of this communication is direct. A change in one molecule's structure can trigger a reaction in another, like a relay baton passed from runner to runner. Some of it is indirect. Small signaling molecules diffuse through the cell, binding to specific targets and altering their behavior. This internal conversation allows the cell to maintain homeostasis, a stable internal environment, despite the changing conditions outside. It tells the cell when to grow, when to divide, when to conserve energy, and when to respond to stress. But molecular communication is not confined to the boundaries of a single cell. In multicellular organisms, cells must coordinate with one another to keep tissues and organs working together. This coordination happens through a variety of signaling systems, each adapted to a different scale and speed. One of the fastest forms of communication is electrochemical signaling, used by nerve cells. Here, changes in ion concentrations across the cell membrane create tiny electrical pulses, which travel rapidly along the nerve's length. At the end of the nerve cell, these electrical signals are converted into chemical ones. Neurotransmitters are released into the narrow gap between cells, where they bind to receptors on the next cell and trigger a new signal. Another widespread form is hormonal signaling. Hormones are chemical messengers, produced in one part of the body and carried through the bloodstream to affect cells elsewhere. Some hormones act quickly, like adrenaline, which primes the body for rapid action. Others act more slowly but with lasting effects, like growth hormone or thyroid hormone, which influence metabolism and development over time. Not all communication travels long distances. In paracrine signaling, cells release molecules that act only on nearby neighbors. This local signaling helps coordinate the activities of small groups of cells, such as in wound healing, where damaged cells release signals that prompt nearby cells to divide and repair the tissue. In autocrine signaling, a cell sends messages to itself, releasing molecules that bind to receptors on its own surface. This may seem redundant, but it is a way for a cell to fine-tune its own activity, like speaking aloud to remember an instruction. At the molecular level, these signals are received and interpreted by receptor proteins. A receptor has a specific shape that matches a specific signal, much like a lock matches its key. When the signal binds, the receptor changes shape, setting off a cascade of chemical changes inside the cell. This signal transduction pathway can amplify the message, turning a single binding event into a large, coordinated response. One common example is the activation of a protein kinase, an enzyme that adds phosphate groups to other proteins, changing their activity. This phosphorylation can turn proteins on or off, altering everything from gene expression to metabolic rate. In many cases, signals are short-lived. Once their job is done, the signaling molecules are broken down or removed, and the cell returns to its baseline state. This prevents the system from staying active too long, which could lead to harmful overreactions. Molecular communication is not just about transmitting instructions. It is also about making decisions. Cells often receive multiple signals at once, some promoting growth, others calling for restraint. The cell's response depends on how it integrates these messages, weighing their strength and importance in the context of its current needs. This ability to integrate information is vital to the health of the organism. In the immune system, for example, cells must respond strongly to genuine threats like viruses or bacteria, but they must also avoid attacking the body's own tissues. A failure in communication can lead to autoimmunity, where the immune system mistakenly targets the self, or to immune suppression, where threats go unchallenged. In development, molecular communication guides cells to their proper places and roles. Chemical gradients, gradual changes in the concentration of signaling molecules, help cells determine where they are in the growing embryo and what type of tissue they should become. Even in adulthood, molecular messages constantly shape our lives at the smallest scale. The sensation of hunger, the repair of a cut, the adaptation to exercise, the regulation of sleep, all depend on precise biochemical exchanges. And molecular communication is not only a feature of multicellular organisms. In bacteria, for example, a process called quorum sensing allows single-celled organisms to measure their population density by releasing and detecting specific signaling molecules. When enough bacteria are present, they can act together, forming biofilms, producing light, or releasing toxins, behaviors that would be ineffective for a single bacterium acting alone. Across all life, from the simplest microbe to the most complex mammal, molecular communication ensures that the parts act in concert with the whole. It is a language without words, spoken in the shapes and charges of molecules, in the fleeting touches between proteins, in the subtle diffusion of signals through the spaces between cells. In our next step, we will explore how this communication plays a role in biochemical cycles, the great recurring pathways that move matter and energy through living systems and connect all life to the earth itself. These cycles are the long, slow conversations between life and its environment, conversations that have been ongoing for billions of years. 18. Biochemical cycles. All living things are made from the same fundamental elements: carbon, hydrogen, oxygen, nitrogen, phosphorus, sulfur, and a handful of others. These elements are neither created nor destroyed within the biosphere. Instead, they move continuously through living organisms, the air, the water, and the soil in great recurring patterns known as biochemical cycles. These cycles are nature's way of recycling the raw materials of life. They link the smallest cell to the largest forest, the breath of an animal to the leaf of a tree, the deep ocean to the highest mountain peak. Through them, The molecules that make up your body today may once have been part of another organism centuries or millennia ago. One of the most familiar of these is the carbon cycle. Carbon is the backbone of organic molecules, and its movement through the biosphere is central to life's chemistry. In the atmosphere, carbon exists mostly as carbon dioxide. Plants, algae, and certain bacteria take in this carbon dioxide during photosynthesis, using sunlight's energy to build glucose and other organic compounds. Animals then eat these plants or other animals that have eaten plants, incorporating the carbon into their own bodies. When plants and animals respire, they release carbon dioxide back into the atmosphere as a byproduct of cellular respiration. When they die, decomposers break down their remains, returning carbon to the soil and air. Some carbon is stored for long periods in fossil fuels, sediments, or ocean waters, slowly re-entering the cycle over geological time. This exchange of carbon between living things, the air, and the Earth's crust has been ongoing for billions of years, shaping the climate and the composition of the atmosphere. The oxygen cycle is tightly linked to the carbon cycle. Photosynthesis not only incorporates carbon into organic molecules, it also releases oxygen into the atmosphere. This oxygen is essential for aerobic respiration, which in turn produces carbon dioxide, feeding back into photosynthesis. The two cycles are like partners in a continuous dance, each depending on the other to keep moving. Another key pathway is the nitrogen cycle. Nitrogen is a vital part of amino acids, nucleic acids, and other biomolecules. Although nitrogen gas makes up the majority of Earth's atmosphere, most organisms cannot use it in that form. It must first be fixed into reactive compounds such as ammonia, nitrates, or nitrites. This is done by certain bacteria, some of which live freely in the soil, while others form symbiotic relationships with the roots of plants like legumes. Plants absorb these nitrogen compounds and use them to build proteins and nucleic acids. Animals obtain nitrogen by eating plants or other animals. When living things excrete waste or die, decomposers break down their nitrogen-containing molecules, returning nitrogen to the soil. Other bacteria then convert some of it back into nitrogen gas, completing the cycle. The phosphorus cycle moves more slowly, but it is no less important. Phosphorus is a key part of DNA, RNA, and ATP, the molecule that carries energy in cells. Unlike carbon and nitrogen, phosphorus has no significant gas phase. It moves mainly through rocks, soil, water, and living things. Over long spans of time, phosphorus is released from rocks, through weathering, taken up by plants, and passed along food chains. Eventually, it returns to the soil and sediments, where it may again be locked away for thousands of years. The water cycle is the most familiar of all. Water evaporates from oceans, lakes, and rivers into the atmosphere, condenses into clouds, and falls back to the earth as rain or snow. Along the way, it flows through rivers, seeps into the ground, and moves through living organisms. Inside the body, water carries nutrients, removes waste, and provides the medium for all biochemical reactions. Its constant movement connects every ecosystem, linking the world's climates, landscapes, and forms of life. These cycles are not separate. They are woven together. The carbon and oxygen cycles are coupled through photosynthesis and respiration. The nitrogen cycle interacts with the carbon cycle through the growth and decay of plants. The water cycle influences all of them, carrying nutrients through soils and rivers, connecting land and sea. The balance of these cycles is delicate. Small changes in one can ripple through the others, altering the availability of vital elements. For example, excess nitrogen from fertilizers can flow into rivers and lakes, triggering algae blooms that deplete oxygen and disrupt aquatic ecosystems. Large-scale changes in the carbon cycle, such as the burning of fossil fuels, affect not only the atmosphere's composition, but also the climate, which in turn influences every other cycle. From a biochemical perspective, these cycles remind us that life is not isolated from its environment. The atoms that make up a living organism are borrowed from the world around it and will one day return. Every breath, every sip of water, every bite of food is part of a continuous exchange between life and the earth. In the stillness of a forest, the quiet growth of a plant is tied to carbon pulled from the air, nitrogen drawn from the soil, phosphorus from the rocks, and water from the rain. In the open ocean, microscopic plankton are doing the same, linking the chemistry of the sea to the chemistry of the sky. From here, our exploration will turn inward again, moving from the grand planetary scale back into the architecture of the cell itself. In the next part, we will explore cellular structures and organelles, the compartments where biochemistry happens, each with its own unique role in sustaining life. 19. Cellular structures and organelles. If a cell is the smallest unit of life, then its internal structures are the rooms, workspaces, and storage areas in which life's chemistry takes place. These specialized compartments, known as organelles, give the cell both organization and efficiency. By separating different biochemical processes into distinct areas, the cell ensures that each task can be carried out under the conditions best suited for it. Cells come in two main varieties: prokaryotic and eukaryotic. Prokaryotic cells, found in bacteria and archaea, are simpler in structure. They lack a nucleus, and most of the membrane-bound organelles found in more complex cells, but they are still highly organized. Their genetic material floats in the cytoplasm, and their biochemical processes occur within the cell membrane and the surrounding fluid. Eukaryotic cells, found in plants, animals, fungi, and many protists, are more elaborate. Their internal membranes create distinct compartments, each with a specific role. These organelles work together, like different departments in a smoothly running factory. At the center of a eukaryotic cell is the nucleus, the control room. Surrounded by a double membrane called the nuclear envelope, it houses the cell's DNA, keeping it safe and organized. Within the nucleus is the nucleolus, a dense region where ribosomal RNA is produced and assembled into ribosome components. The nuclear pores in the envelope allow mRNA and other molecules to pass in and out, connecting the flow of information between the nucleus and the rest of the cell. Surrounding the nucleus is the endoplasmic reticulum, or ER, a network of membranes that serves as one of the cell's main production lines. The rough ER, studded with ribosomes, is where proteins destined for secretion or for membranes are synthesized. The smooth ER without ribosomes has roles in lipid synthesis, detoxification, and the storage of calcium ions. From the ER, newly made proteins and lipids often travel to the Golgi apparatus. This organelle functions like a packaging and distribution center. It modifies molecules, adds chemical tags that direct them to their destinations, and sorts them into vesicles, small membrane-bound packages for transport. The mitochondria are the cell's power plants. With their own DNA and ribosomes, they produce ATP through cellular respiration, converting the energy stored in food molecules into the form the cell can use. Mitochondria have a double membrane, with the inner membrane folded into cristae that increase the surface area for energy production. In plant cells, there is an additional powerhouse, the chloroplast. These green organelles capture sunlight and convert it into chemical energy through photosynthesis. Like mitochondria, chloroplasts have their own DNA and ribosomes, and they are surrounded by a double membrane. Inside, stacks of thylakoid membranes contain chlorophyll and the machinery needed to harvest light. Scattered throughout the cytoplasm are the ribosomes, the protein factories of the cell. They can be free-floating, producing proteins for use inside the cell, or attached to the rough ER, making proteins for membranes or export. The lysosomes are the recycling and waste processing centers. They contain powerful enzymes that break down old organelles, large molecules, and even engulfed foreign particles. In plant cells, a similar role is often played by the central vacuole, which also stores water, nutrients, and waste, and helps maintain the cell's shape by creating internal pressure against the cell wall. The peroxisomes are small but important. They break down fatty acids and detoxify harmful substances, producing hydrogen peroxide in the process, which they then quickly neutralize. The cell's internal framework is provided by the cytoskeleton, a network of protein filaments that gives the cell its shape, anchors organelles in place, and provides tracks along which materials can be transported. The cytoskeleton is dynamic, able to reorganize as the cell moves, grows, or changes shape. In plant cells, a strong cell wall surrounds the plasma membrane, providing support and protection. Composed mainly of cellulose, the cell wall allows the plant to maintain its structure, even without the rigid skeleton that animals possess. Finally, the cell membrane or plasma membrane forms the boundary between the cell and its environment. Composed of a phospholipid bilayer with embedded proteins, it regulates what enters and leaves the cell, allowing nutrients in, letting waste out, and controlling the flow of signals between the inside and outside world. Each of these structures has its own role, but none work in isolation. The nucleus sends out instructions. The ER and ribosomes build proteins. The Golgi packages them. The mitochondria power their transport, and the cytoskeleton guides their movement. The lysosomes and peroxisomes keep the system clean and functional, while the membrane manages communication with the outside world. In this way, a cell is less like a random collection of parts, and more like a well-designed city, with centers of governance, factories, power plants, transport networks, recycling facilities, storage depots, and borders that define and protect it. In our next and final section, we will step back and look at the integration of biochemical systems, how everything we've explored, from molecules to organelles to cycles, fits together into the unified, ongoing miracle of life. 20. Integration of biochemical systems. Over the course of our journey, we have explored life from its smallest molecular building blocks to the grand cycles that link all living things to the earth itself. We have seen how carbon, nitrogen, water, and energy flow through organisms, how proteins, lipids, carbohydrates, and nucleic acids carry out their diverse but interconnected roles, how organelles within the cell act like coordinated departments in a well-run city. But the true beauty of biochemistry lies in how all of these pieces fit together, forming a single, seamless system in which every part depends on every other. At the foundation are the chemical elements and the molecules they form. These molecules are not static. They are in constant motion, colliding, reacting, and transforming into new shapes. The language of chemistry: covalent bonds, hydrogen bonds, ionic attractions, allows atoms to form the sugars, fats, proteins, and nucleic acids that make life possible. These molecules, in turn, do not simply float aimlessly. Inside the cell, they are organized into pathways, chains of reactions, in which the product of one step becomes the starting point for the next. Enzymes guide each step with precision, making sure reactions happen quickly enough and in the right order. The pathways themselves are woven into the larger web of metabolism, balancing the breakdown of molecules for energy with the building of new molecules for growth, repair, and adaptation. This balance is not rigid but dynamic, adjusting to the availability of nutrients, the demands of activity, and the signals received from other cells. Energy flows through this system like a quiet, steady current. ATP is generated in mitochondria through cellular respiration, fueled by the glucose made available through digestion, or, in plants, through photosynthesis. This ATP powers the work of the cell-moving molecules across membranes, building complex structures, contracting muscle fibers, sending nerve signals. Without the constant renewal of ATP, even the simplest processes would quickly cease. Information also flows through the system. DNA holds the instructions for making every protein the organism needs. Through transcription and translation, these instructions are read and carried out, ensuring that the right proteins appear at the right times and in the right places. Regulation at every step prevents waste and maintains balance, allowing the organism to adapt to both its internal state and its surroundings. Communication between cells coordinates the activities of tissues and organs. Hormones, neurotransmitters, and local signaling molecules carry messages that influence metabolism, growth, and repair. This molecular communication ensures that no part of the organism works in isolation. The liver adjusts its glucose release in response to the needs of muscles. The brain triggers the release of hormones that affect the immune system. The skin signals for repair when it is injured. Beyond the individual organism, life participates in the biochemical cycles that move elements and energy through the biosphere. The oxygen you exhale may be used by a tree to release the glucose that will one day fuel another breath. The nitrogen in your proteins may, in time, return to the soil to nourish a plant, which will pass it along to another life form. Every living thing is a temporary caretaker of the elements it holds, passing them along to others in an unending cycle. The cellular structures and organelles we explored earlier make this integration possible. Each one has its own role, but none could fulfill its purpose alone. The nucleus provides the genetic instructions. The endoplasmic reticulum and ribosomes build proteins. The Golgi apparatus directs them to their destinations. Mitochondria and chloroplasts supply the energy. Lysosomes and peroxisomes recycle materials. The cytoskeleton shapes and organizes the whole. The cell membrane maintains the delicate balance between openness to the environment and protection from it. In essence, biochemistry is the study of life's interconnectedness. It reveals that there is no sharp line between structure and function, between energy and matter, between one process and another. All are part of the same ongoing flow. The glucose made in a leaf during photosynthesis may be broken down in an animal's cell to make ATP, which may then power the folding of a protein encoded by DNA, which may in turn act as an enzyme in a metabolic pathway, the products of which may become building blocks for another living thing. This seamless integration gives life its resilience. When one pathway slows, others can compensate. When resources are scarce, energy is conserved, and priorities shift. When conditions change, the system adapts, sometimes within minutes, sometimes over generations. As we step back from the details, we see the whole picture. Life is a continuous exchange of matter, energy, and information, carried out by countless molecules working in harmony. The complexity is immense, but it is also elegant. Every heartbeat, every breath, every moment of growth or repair is the product of this intricate collaboration. And so, the study of biochemistry is not just the study of molecules. It is the study of the quiet unity of life, the way that every cell, every organ, every organism is part of a larger story written in the shared language of chemistry. It is a reminder that, despite the apparent diversity of life on Earth, we are all built from the same materials, powered by the same energy, guided by the same molecular principles. From the smallest bacterium to the tallest tree, from the humblest algae drifting in the sea to the most complex human brain, the same biochemistry sustains us all. And in its gentle, ceaseless work, it keeps the living world turning day after day, cycle after cycle, generation after generation.